Thank you so much, Matt. My name is Jordan Green. I'm scientific co-founder and CTO at Dome Therapeutics, and I'm excited to share with you all of our work on next-generation nucleic acid nanomedicines to treat CNS disorders and end brain cancer. And so we're leveraging innovative technology that can safely and effectively deliver nucleic acids, flexible on the different cargos, to target cells. And what we can do is turn on or off genes and be able to program cells. And our initial focus is on glioblastoma. This is a large market in the US and around the world. And it's an initial demonstration of this technology that we believe will also be very useful for CNS disorders more broadly. And we're leveraging competitive advantage of having this development of the technology that's been more than a decade and more than $10 million in grant funding to Hopkins and Mayo to develop this technology that improves efficacy, improves safety, and reduces costs. Glioblastoma is a terrible disease in need of new treatments. It's the most common type of adult primary brain tumor. It's half of all malignant tumors. It's treated with surgery, radiation, chemotherapy, a very aggressive therapy, and yet median survival is just over a year, and a 5-year survival is under 5%. And the challenge is that there's resistance and recurrence. And so Dome has designed a technology to solve this challenge. Glioblastoma as a first indication is interesting both because it's a large unmet need that there need to be new help for these patients that are suffering. It's also, though, $1 billion market in the US, more than $2 billion worldwide. And it's an orphan disease, and so there's advantages there in terms of the FDA regulatory pathway. And from the payer side, it's the most expensive to treat of any cancer. It's $150,000 in treatment costs in that first year, and that the patients still die shortly after that. The standard of care is insufficient. Radiation therapy has been around a long time. It's expensive. Surgery and radiation can extend survival to about a year. The biggest advance is actually the glial wafer, which is a technology co-developed with Hopkins, that is a local therapy following surgery, and that helps extend survival a bit longer, but it's not a cure. And then the other technologies also are just very modest survival benefits. And so we're building on the approach to have a local therapy, but then one that can dramatically improve the outcomes. And what the change is, and the new paradigm that we've discovered and we're innovating, is to look at cancer stem cells. These unlock a new treatment path. And so the clinical problem is this near universal recurrence. And my clinical co-founder, Dr. Alfredo Quinones Hinojosa, Dr. Q, this is from one of his patients when he was here at Hopkins who'd present with the glioblastoma. You see all that white contrast, that's the GBM, receiving excellent care with surgery and post-op care. It looks like all the GBM is gone, but 2 months later, looking at the MRI, it all comes back. And this is what makes the disease so devastating. And what it is that cancer stem cells are the seeds that then regrow this tumor. They're responsible for the relapse, the metastasis, and the resistance to conventional therapeutic approaches. This is our leadership team of key personnel at Dome. Dr. Q, who I mentioned, was at Hopkins till full professor and then got recruited to Mayo Clinic, Florida to direct their neurosurgery program there. He's an internationally known neurosurgeon with a lot of expertise in CNS disorders, including GBM. And our broader team is a team of proven innovators and entrepreneurs that have the expertise and talents for our next steps. The central technology challenge is identifying the cancer stem cells and then understanding the biological mechanisms that can turn them off. There's a need for improved technology for safe and effective delivery. And if you look at the technologies out there, viruses can be very effective, but they have challenges with safety, with manufacturing, and other challenges. Lipid nanoparticles solve some of these, but they're not as effective as the viruses generally, and they also have challenges with targeting. And traditional polymers aren't very effective or safe. But what we've done with Dome is develop next generation nanoparticles, biodegradable polymeric nanoparticles, PNPs, that are ideally suited for RNA and DNA delivery to a range of different cells and tissues. And so these technologies we've validated as having viral-like efficacy, but also being very safe. They degrade in water in just hours. We can have different size cargoes from oligonucleotides to large plasmids, and this can be scaled up and manufactured very easily. We've designed these nanoparticles, again, it's leveraging 10 million in funding, to be able to go through each of these steps in delivery in a very efficient way. Instead of needing multiple components, it's just the one polymer mixed with the nucleic acid. The primary amines and secondary amines bind and protect that nucleic acid, protect it from degradation outside the cell, and enable cell-specific delivery into our targets. Just like when we eat food, we digest what we eat. When cells internalize these particles, they degrade what's inside in these endosomes and lysosomes. But what we've designed with these polymers is a very high density of ionizable amines that buffer the endosomal compartment and lead to very efficient release and escape out of the endosome into the cytosol where they work. And then the polymers have these ester linkages throughout their structure that rapidly degrade with a half-life on the order of about 5 hours. This gets the biomaterial out of the way so that it doesn't cause toxicity or incorporate with the cell, and it efficiently releases the cargo that can then have its effect. So Dome's nanoparticle nanomedicine platform enables engineering of cancer stem cells, and I'll show you data next on how that works, but other cells too. And so it's a biodegradable, safe, and efficient platform that's also very scalable and low cost. We can turn on or off target genes in the brain as well as elsewhere, and we have a broader platform of cell and gene therapy also throughout the body. So here's some of the data. The Dome's nanoparticles can kill brain cancer stem cells. It spreads through a tumor. The nanoparticles are about 100 nanometers in size. They're able to then turn off the programming of what makes a cancer stem cell a stem cell, so they can then be treated. What we find with survival is instead of bending the curve, we see long-term survival with the tumors that then melt away, the next slide, and then we see 70% long-term survivors where they don't have tumors anymore. And so when we look at their brains afterward, all the tumor has been gone, where in the control cases it's very large. Moreover, we see if we do a lower dose of the particles, we can see how it's also synergistic with radiation. So if you do the plus is radiation, you don't see that much of effect with radiation because the resistance of these cancer stem cells to radiation, but with nanoparticle treatment, you see a reduction in the tumor volume, and then with radiation, it's synergistic. Or just with the combination microRNA, we see the tumors fully eliminated. We've looked at the manufacturing. We can scale this up. We can store it stably. So the particles can stay stable at -20 for years, or in the fridge, they're stable for many months. And this is unlike a lot of other nucleic acid technology that needs different kinds of cold chain for distribution and use. We can use our flexible gene and cell therapy platform to look at many different CNS disorders, and we have other indications in our pipeline. We've recently received a TEDCO MII award to move this from the rodents to dogs as a large animal model, and so this is ongoing now. And in addition to being a key model for patients as a large animal model that develops brain tumors, what we've also found is that we can look at the veterinary medicine pathway to treat patient dogs, pet dogs, that have brain cancer, and that it would take just 25 dogs to show the reasonable proof to the FDA that then we can market and sell to the pet market, which can raise a lot of revenue for then the clinical development as well. We understand our commercialization pathway, so what we're looking for is a seed to IND, which would be able to develop the canine platform to FDA conditional approval and to sell, as well as developing the human version through GMP and IND-enabling PK and tox studies for then the subsequent human studies. For the dog market, we just need to show survival that can last 3 months or more. And then for the human patients, with this technology that we saw with the animal models that did long-term survival, we'd want to show durability of 20 months or more. So thank you so much for your attention. I hope I've shown you how we're so excited at Dome about innovating these new treatments for brain cancer and neurological disorders. And I'd be happy to answer any questions.